Welcome to Tomorrow Unveiled, the program dedicated to delivering the definitive update on the future of human health and technology. Every year, we talk about breakthroughs in longevity science,
Right.
but 2025 is really marking a pivotal shift. It's one that moves us beyond, just fighting single diseases. We're heading towards this high-stakes precision engineering of, superior biological function.
Mhm.
For you, the listener, this means the goal isn't just about extending your chronological lifespan anymore. It's about maximizing what we call health span, functional life extension. Our research team has been digging deep, and they've extracted and cross-corroborated the most crucial nuggets of knowledge from all the latest global breakthroughs. And the message is it's resounding. The next decade of aging science is fundamentally immunological and informational.
That's the critical distinction we need to make right out of the gate. For decades, the public conversation about longevity was all about finding a magic bullet, right?
Like a single pill.
Exactly, a single pill to slow down this generalized decay. Now, we're documenting the transition from these blunt systemic treatments to highly specific, almost mechanized corrections of cellular entropy. It's a quantum leap, a huge one, because we're learning how to reset the core programs that drive the aging process itself rather than just, slowing down the symptoms.
So we're getting at the source code.
We are. The information we compiled shows a powerful consensus emerging. It's right at the intersection of three fields. First, leveraging the adaptive immune system for continuous biological maintenance.
Okay.
Second, achieving precise cellular reprogramming to restore youthful function. And third, using these next-generation biomarkers to measure and optimize every single intervention with just unprecedented accuracy.
So this isn't just theory anymore.
No, not at all. These are translational platforms. A lot of them are already filing investigational new drug applications, INDs, for human trials in the very, very near future.
Okay, let's unpack this shift in strategy then. Let's start with probably the most destructive force in aging, senescent cells. For years, the conversation was all about classic senolytics, small molecules like dasatinib and quercetin or D plus Q. The idea was to clear out these zombie cells.
Right.
They've stopped dividing, but they refuse to die. And instead, they just secrete this nasty pro-inflammatory cocktail. The SASP,
SASP, yeah.
D plus Q was a good start.
Yeah.
But the problem was always twofold, right? They weren't totally specific, so you had off-target effects, and you needed constant repeated dosing to keep that senescent burden down.
Exactly. And the shift we're seeing now is it's decisive. We're moving from small molecule chemistry to advanced immunology. The problem with pills, you see, is that they treat the body as a static system.
Okay.
The future treats the body as the dynamic battlefield, and on that battlefield, the immune system is the perfect weapon, provided you train it properly.
So that's why you're saying senolytics are becoming vaccines.
That's it. Senolytics 2.0.
Senolytic vaccines. That concept just sounds incredibly advanced. Can you walk us through the mechanism of this breakthrough platform, specifically the one the research team is tracking, Senovax? How do you vaccinate against your own aged cells?
Well, the core mechanism of the Senovax platform, which was developed by some real pioneers in the field, is its genius and its simplicity. It's all about bypassing that need for chronic drug administration.
Right, taking a pill every day.
Yes. By programming the immune system to do the work permanently. The process involves identifying these specific tiny protein fragments. We call them cryptic antigens. And they only appear on the surface of senescent cells. You don't find them on healthy cells.
So they're like a little red flag.
They're a perfect red flag.
Yeah.
The vaccine then uses these specialized immune cells called dendritic cells to capture these antigens and then present them to the T cells.
The T cells are the body's assassins, right?
They are. And by presenting the antigen, the dendritic cells are essentially giving the T cells a most wanted poster. It's got the mugshot of every zombie cell in your body. And this turns the entire adaptive immune system into a precision senolytic weapon, capable of continuous systemic clearance.
The implication there is it's enormous. It means eliminating compliance issues with pills. And you could potentially get a much higher clearance rate system-wide. When we look at the data, and I have to ask you about the headline-grabbing figure, are these immunological approaches just theoretically better, or are the preclinical results truly showing a massive step up?
The preclinical data, and this is compiled from several reports by the research team, are Well, they're quite frankly astonishing. They claimed extensions in lifespan and health span greater than 100% in certain short-lived animal models.
Okay, let's put a pin in that for a moment.
Yeah.
Double the lifespan. That is a truly disruptive claim. What are the necessary caveats we need to put around that 100% figure, and what does it tell us about the mechanism?
Of course. We have to approach all animal data with extreme caution, especially when you see numbers like that.
Sure.
Mice aren't humans. Their aging pathways are distinct. However, the magnitude of the effect, regardless of the species, it strongly suggests that immunological control of senescence is just vastly more potent, much more than the transient chemical inhibition you get from the old Senolytics.
So the immune system is just better at the job.
It indicates that the immune system, once you aim it properly, achieves this state of near-perfect continuous surveillance and destruction of senescent cells. It's a level that pills simply cannot match. If even a fraction of that efficacy translates to humans, it represents a profound shift.
And beyond just pure longevity, the sources also highlighted a critical secondary benefit, one that plays into the commercial strategy.
Yes, the broad anti-tumor efficacy. See, senescent cells don't just sit there. They actively create this permissive, supportive microenvironment for new tumors, a tumor-supportive niche.
So they're helping cancer grow.
They are. And eliminating these senescent cells showed dramatic reductions in various cancer models. We're talking lung, breast, brain, skin, even pancreatic tumors.
That's a very broad effect.
It is. And this broad anti-cancer effect is key to their translation strategy. An investigational new drug application, an IND, has already been filed for the Senovax platform. And it's initially targeting non-small cell lung cancer.
That's a brilliant strategic move.
It is. It positions the long-term use of this vaccine as a form of immunological maintenance, like a booster shot for longevity, by first treating it as a potent preventative cancer therapy. It helps them get past all the regulatory hurdles for an anti-aging indication, which is almost impossible.
That is incredibly smart. I mean, using cancer as the regulatory vehicle just illustrates how fundamentally this science is reshaping the entire pharmaceutical landscape. But this immune approach isn't just limited to these broad-spectrum vaccines, is it? We are also seeing highly specific engineered cell therapies being deployed.
That's right, and this is where the specificity gets truly futuristic. We are talking about engineered T cells, specifically CAR T cells. Historically, CAR T cell therapy has been a really complex, personalized cancer treatment.
Right, where you take a patient's own T cells out, reprogram them.
Exactly. Reprogram them to attack cancer and then reinfuse them. Now, that same technology is being precision aimed at specific senescent cell populations in organs that are most vulnerable to aging.
Which organ are they targeting first, and what's the specific marker they're hunting for?
The target is the aging gut. And the specific marker is something called the urokinase plasminogen activator receptor, or UPAR.
UPAR.
These UPAR-positive senescent cells, they just disproportionately accumulate in aging intestinal tissue. And the aging gut is really ground zero for chronic systemic inflammation because it suffers from poor barrier function, leaky gut, and reduced intestinal stem cell regeneration.
So what happens when you send in the CRT cells?
The research showed that a single infusion of these anti-UPAR CAR T cells in animal models achieved just profound results. It reversed age-associated intestinal dysfunction, which led to significant functional benefits.
Like what?
Well, it improved the gut barrier, it dramatically enhanced the regeneration of intestinal stem cells, which are crucial for renewing the gut lining, and it optimized the gut microbiome composition just by removing those inflammatory drivers.
That's a massive systemic impact from targeting a really localized issue. I mean, the gut is so essential to metabolism, nutrient absorption, immunity. But the complexity of CAR T usually means very high costs and logistics. Is this a durable intervention?
The durability is the selling point here. Unlike pills that need constant top-ups, the research team highlighted data showing that a single prophylactic dose given to younger mice provided sustained protection for over 15 months, which suggests really long-term health span gains from an early intervention. The beauty of immune memory is that once those T cells are trained and established, they stick around.
They remember the enemy.
They do. And this fundamentally moves the conversation from chronic treatment to true immunological maintenance, offering durable protection against a core hallmark of aging.
That transition from continuous clearance to durable long-term repair, it leads us to the ultimate goal. Not just stopping decay, but actively reversing age at the tissue level. We're moving from immunological maintenance to true biological rejuvenation. Here's where it gets really interesting, diving into the management of biological information itself.
This field is rapidly maturing, and it's under the banner of partial epigenetic reprogramming, or PER. To understand PER, we have to start with the foundational information theory of aging.
Okay.
This theory, it posits that aging isn't primarily caused by damage to the DNA hardware itself. It's the loss of information in the epigenetic software layer.
The software, not the hardware.
Exactly. The epigenome is the set of chemical tags like DNA methylation patterns that tell the underlying DNA which genes to express and which to keep silent. As we age, that software gets corrupted, the methylation patterns degrade, and cells, well, they forget how to function optimally.
So if DNA is the original pristine recipe book, the epigenome is like the handwritten instruction manual on how to use it. And over time, the ink is bleeding, the pages are stuck together, and the instructions are getting garbled.
That is a perfect analogy. And the PER strategy seeks to restore those youthful methylation patterns. This is achieved using a very precise subset of the famous Yamanaka factors, Octet, SOX2, and KLF4, often referred to as OSK. They essentially reboot the cell's corrupted software.
Now, this is where the critical nuance comes in. We know the original 4 Yamanaka factors can completely revert any adult cell back to a pluripotent embryonic stem cell state, which sounds powerful, but it's a massive cancer risk because those cells are totally uncontrolled. So how does PER manage that risk?
That is the safety lock, and it's the genius of the PER approach. Using all 4 factors is highly oncogenic, very unpredictable. The PER strategy uses only the OSK factors and critically administers them for a very short, precisely timed pulse, a transient expression.
So it's just a quick flash.
A quick flash. This allows the cell to partially reprogram and restore that youthful DNA methylation signature, effectively turning back the biological age clock. But without achieving full cellular dedifferentiation, the cell restores its youthful instructions, but it remains a liver cell or a retinal cell. Its identity is intact, and that eliminates the immediate risk of tumor formation.
That safety lock is what makes this concept translational. It moves it from theoretical science to clinical reality. What's the timeline on this, and what are the first human targets?
The intelligence we compiled confirms the PER platform is on track for first-in-human trials by Q1 2026.
That's soon.
It is. They're targeting high-need, specific organ failures. Candidate ER100 is focused on optic neuropathies, reversing degeneration in the retina, and candidate ER300 is focused on severe liver disease, specifically metabolic dysfunction-associated steatohepatitis, or MISH.
MISH is more than just a fatty liver, isn't it?
Yeah.
Could you define MAH for us and explain why the liver is such a perfect testing ground for a systemic rejuvenation therapy?
Sure. MH, which was formerly known as NASH, isn't just fat accumulation. It's a disease characterized by fat, inflammation, and progressive scarring, or fibrosis, all driven by chronic metabolic dysfunction.
And it's becoming more common.
These are the master cells that produce all the blood and immune cells in your body. As they age, they rapidly lose their ability to regenerate. And that leads to severe immunosenescence, an aged, dysfunctional immune system, and eventually age-related blood failure.
Okay, so what goes wrong with them?
The research team identified the precise mechanical drivers of aging in HSCs, which is just fascinating. There are 2 key factors. The first is lysosomal hyperactivation.
For lysosomes, those are the cell's recycling centers.
They are, and they become hyperacidic and overworked, which essentially stresses the cell's entire maintenance system. The second factor is increased nuclear tension, which is regulated by a pathway called RhoOA.
What exactly is the RhoA pathway, and how does that relate to the nucleus stiffening up?
RhoOA is a critical regulator of the cytoskeleton, which is the internal scaffolding of the cell. You can think of it as controlling the cell's internal pressure and stiffness. In aging HSCs, increased RhoOA activity leads to greater tension and rigidity. It literally stiffens the nuclear envelope, the membrane that surrounds the DNA. The sources indicate that this physical stiffness mechanically restricts the cell's ability to access the genetic code it needs for rapid, youthful regeneration.
So the cell's factory is locked down by its own internal tension.
Exactly. We have a biological mechanism, hyperacidity, stressing the machinery.
And a physical mechanism, stiffness,
Yeah,
locking the factory door.
precisely. But knowing the mechanical cause provides a precise fix. Researchers demonstrated that using specific drug inhibitors to either suppress that lysosomal hyperactivity or inhibit RhoOA restored the regenerative capacity of the old HSCs.
How did they do that?
By reducing the acidity and relaxing that nuclear tension, they restored mitochondrial function and dramatically enhanced the ability of these old stem cells to produce new functional blood.
The results must have been pretty clear.
Oh, they were highly quantifiable. Ex vivo treatment of old blood stem cells with these interventions boosted their ability to form new functional blood in animal models by more than eightfold.
Eightfold.
This is foundational research that gives us clear pharmacological targets for drug development. It suggests a pathway to prevent age-related immune failure and enhance the immune response in the elderly dramatically.
So, we have the tools for deep immunological clearance and fundamental epigenetic repair moving into clinical trials. The next critical layer is precision. How do we manage the complex metabolic environment that drives most age-related diseases? And how do we use powerful new tools like AI to make these interventions precise and rapid?
The landscape of drug discovery is, well, it's no longer a slow-moving, random screening process. It's now an engineering challenge solved at high velocity. And that's thanks largely to generative AI platforms. The velocity and novelty provided by the AI drug discovery engine are just compressing the traditional pharmaceutical timeline. Traditionally, it might take, 7 to 10 years to move from identifying a biological target to creating a viable molecular candidate.
A long time.
A very long time. Generative AI is collapsing this into 12 to 18 months, from target identification to candidate nomination.
How does the AI actually achieve that velocity? Is it just scanning existing molecules faster, or is it truly inventing new drugs?
It's the latter. It's inventing. These systems hallucinate novel molecular structures.
Hallucinate? I like that.
They're trained on these massive data sets of chemical properties, toxicity profiles, biological targets. They act as generative adversarial networks, or GANs, and they can design molecules optimized for multiple competing parameters all at the same time.
So they're looking for the perfect fit.
The perfect fit for the biological target, the ideal oral bioavailability, minimal toxicity, and maximal efficacy. They're creating these perfect fit molecules that have never existed in nature, targeting newly discovered aging pathways.
What's the main application right now?
The primary application is in cardiometabolic assets. Because the AI moves so fast and creates such safe, optimized molecules, it's making it financially viable to develop drugs for preventative indications, what we call geroprotectors.
I see.
These target the root pathways of metabolic syndrome and fibrosis, which were previously considered too financially risky for traditional pharma timelines. The reduced costs and increased predictability of development. It just changes the economic equation entirely.
Yeah, that velocity is a game changer. It means the next generation of longevity drugs are already being engineered right now in parallel with the cell therapies we just discussed. But speaking of metabolism, we have to talk about the massive public interest in the GLP-1 longevity question.
Absolutely. The GLP-1 receptor agonists, these are the drugs famous for inducing dramatic weight loss, the ones you see in all the headlines.
Yes.
They're now being analyzed for a much deeper anti-aging mechanism that appears to be completely independent of the weight loss they cause.
Wait, so you're saying that the longevity benefits come even if you're not obese or diabetic?
That is the crucial discovery that was corroborated by the research team. Studies on aged mice treated with low-dose GLP-1 agonists showed significant improvements in markers of physical performance grip strength, rotarod tests.
Without requiring any weight loss.
Exactly. This completely decoupled the core metabolic effect, the weight loss, from the systemic restorative effects.
What's happening on a molecular level?
Well, advanced genetic and molecular analyses showed that the drug reversed age-related gene expression and DNA methylation patterns across multiple tissues. The animal wasn't just thinner, it was molecularly younger.
Wow.
And this points toward a deeper, brain-mediated anti-aging mechanism, suggesting that the GLP-1 receptor, which is highly expressed in the brain, is the central control point.
Where in the brain is this critical action happening?
The data strongly pointed to the hypothalamus.
The brain's control center.
Exactly. For appetite, metabolism, endocrine function. Researchers found that selectively knocking down the GLP-1 receptors in the hypothalamus significantly attenuated or reduced the systemic anti-aging benefits. This suggests GLP-1s are acting on the brain to reset metabolic and inflammatory control systemically. It's a master regulator of biological aging, not just weight management.
This has profound implications for how these drugs are used. If the primary anti-aging benefit is mediated through the brain and doesn't require that massive fat loss that can sometimes lead to muscle wasting and GI distress, then, well, the solution seems obvious: microdosing.
And that is the direction of the clinical shift. The sources indicate a dedicated clinical trial has been initiated to test microdoses, fractions of the standard therapeutic dose used for weight loss, in healthy, non-obese individuals.
And they're not just measuring weight.
No. The trial is specifically measuring longevity metrics and quality of life indicators rather than just the number on the scale.
This does raise a serious ethical and regulatory question, though. We're taking potent, complex drugs designed for serious metabolic disease, and we're repurposing them for healthy populations in a preventative role. What are the potential downsides we need to be watchful of with this kind of brain-mediated control?
The primary concern is long-term, unintended consequence, of course. While the microdoses may avoid the severe GI distress, altering central metabolic control in a healthy body for decades, it carries unknown risk.
So what do we need to look for?
We need meticulous long-term monitoring for things like pancreatic stress or subtle changes in mood and cognitive function, given that hypothalamic involvement. The goal of this trial is to challenge the old regulatory framework to establish that a drug can be a preventative tool for health span. But that requires impeccable safety data, especially when you're expanding use to healthy people.
That's the balance we have to strike, isn't it? Velocity against safety. If you find this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future.
And if you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners.
Okay, so if we're going to use AI to design precision drugs and use cell therapies to partially reverse age, we need equally precise tools. Tools to measure what exactly is happening inside the body. The era of just using your chronological age is obsolete. We need to know, what is your real functional biological age? And maybe more importantly, where is the most damage concentrated?
This is the measurement revolution, and it provides the feedback loop for all the interventions we've just discussed.
Yeah.
The first major leap forward is the validation of organ-specific proteomic clocks.
Proteomic clocks? Okay.
These new biological clocks are fundamentally different from the previous DNA methylation clocks. They're derived from analyzing plasma proteins, the thousands of proteins circulating in your blood from these massive cohorts, including over 43,000 participants in the UK Biobank.
And the innovation is the organ specificity.
Exactly. They calculate the biological age of specific organs, the brain, kidney, liver, lung, heart, and more. It's not just a single generalized biological age for the whole person.
That granularity changes everything. I mean, you could have a biologically young heart from great cardiovascular fitness, but a very old liver because of a poor diet, or an old brain from chronic stress.
Yeah.
How does this organ-specific insight improve prediction?
It reveals that individuals age at dramatically different rates across their organ systems, something we've long suspected but couldn't really prove.
Right.
The prediction power of these clocks is profound because it personalizes risk. Accelerated brain age, for example, which is derived from the signature of proteins in the plasma, strongly predicted future risks of dementia, Alzheimer's risk.
Especially with certain genes.
Yes, especially in individuals with the high-risk APOE4 gene and also cardiovascular events. This offers far more personalized and actionable risk assessment than general health metrics. It allows for hyper-targeted intervention focused on your most vulnerable organ system.
That's amazing.
And complementing these molecular clocks, we have the validation of structural biomarkers that link lifestyle and body composition directly to neurohealth. This is what the research team calls the muscle-fat-brain axis.
The muscle-fat-brain axis. So how does structural imaging, like an MRI, tell you how old your brain is on a biological level?
Well, the information we compiled highlighted these large-scale studies that used AI analysis of whole-body MRIs of thousands of adults. And this established a clear macroscopic link between specific types of body composition and neurological integrity. The AI is trained to correlate body metrics with structural brain changes, like volume loss and white matter integrity.
And what was the key finding?
The key finding was devastatingly clear and specific: high visceral fat.
That's the deep abdominal fat around your organs.
That's the one. It was strongly associated with an accelerated biological brain age. Individuals with more belly fat had brains that looked structurally older, showing measurable atrophy and reduced white matter integrity.
And why is that type of fat so bad?
That specific fat is metabolically active. It secretes pro-inflammatory cytokines that cross the blood-brain barrier and drive neuroinflammation.
So not all fat is created equal. It's the fat deep inside that's the real accelerant for brain aging. But what about muscle mass? Because that's often lost as we accumulate fat with age.
Conversely, high muscle mass was shown to be neuroprotective. Individuals who maintained higher levels of lean muscle had younger-looking brains and preserved white matter integrity.
It's interesting.
And crucially, the sources noted that the fat stored just under the skin, subcutaneous fat, appeared to be metabolically neutral in this context.
It's just there.
It's just there. This validates the hypothesis that muscle acts as an endocrine organ. It secretes these protective molecules called myokines that actively fight the inflammation driven by visceral fat. So this makes whole-body MRI, analyzed by AI, a potent longevity screening tool. It directly validates lifestyle levers, exercise, strength training, diet as measurable proxies for brain health.
So you can actually see the result of your efforts, not just on the scale, but in the measurable age of your brain.
Precisely.
That's the ultimate proof of concept for strength training. Okay, finally, let's talk about inflammation biomarkers that are moving into the clinical utility stage, proving whether these new drugs are actually working.
We are seeing key biomarkers mature into true trial validation tools. Glial fibrillary acidic protein, or GFAP, is a prime example.
GAP.
It's a marker released when astrocytes, these are the star-shaped cells that support neurons, are activated in response to inflammation or injury in the brain. So it's a direct indicator of neuroinflammation.
So how is it being used?
The reduction in plasma GFE is now being used as a primary biomarker validation in neurodegeneration trials. For a drug to be considered successful, it's not enough to show a slight cognitive improvement anymore. It has to prove it's hitting the root cause.
So if GAP levels go down,
If a drug reduces plasma GFAP levels, it proves that the compound is successfully hitting its anti-inflammatory target and creating a measurable physiological improvement in the human brain. This is crucial for moving drugs from treating symptoms to true disease modification.
got it. And what about systemic frailty?
For that, there's another novel inflammation biomarker that helps target it, EDA2R, the ectodisplacin A2 receptor. The research highlighted that its expression increases consistently with age across various tissues, and it's not just a correlative marker of aging, it's a driver.
A driver of what?
EDA2R activates the master regulator of chronic inflammation, the NFE pathway, and that's strongly associated with age-related frailty and sarcopenia, or severe muscle loss. Identifying it gives researchers a precise molecular target to dampen inflammation, potentially reversing frailty at its source.
The granularity is just incredible. We've moved from vaguely measuring inflammation to targeting specific inflammatory arresters, measuring the biological age of every major organ, and using structural imaging to predict neurodegeneration. We are truly entering the era of personalized biological maintenance. So we have immunological clearance, we have epigenetic repair platforms moving to human trials, AI designing perfect molecules, precision clocks measuring every biological system. The science is accelerating beyond our wildest predictions. So what does this all mean for society? And what's the next great frontier in basic science? So what does this all mean?
The acceleration forces us to confront some essential ethical and practical questions, primarily concerning access. The access dilemma is perhaps the most immediate social challenge.
The cost.
The cost. High-cost cutting-edge therapies like customized CAR T-cell treatments or forthcoming gene editing solutions, they could easily carry price tags in the hundreds of thousands of dollars or more.
Which most people can't afford.
Exactly. And this raises the terrifying prospect of a biological caste system where the extremely wealthy aren't just financially better off, but biologically superior, enjoying dramatically longer and healthier lives.
That's a scary thought.
It is. Philosophers and ethicists are debating whether the traditional inevitability of death, the aging status quo, has always served as a sort of universal equalizer that fosters a common humanity. If that equalizer is removed for a privileged few, it risks rupturing our social contract.
So what's the solution?
The ethical debate suggests the solution has to be rapid democratization and cost reduction. Otherwise, the entire field risks a massive public backlash.
And that debate speaks directly to the language the scientific community is using, right?
Yep.
Which is rapidly professionalizing to achieve legitimacy.
Yes, the research team noted a concerted effort by serious researchers to distance themselves from the term anti-aging.
It has a bad reputation.
It does. That term is now associated with, speculative commercial cosmetics and unproven supplements. The field is actively rebranding to rigorous geroscience, or gerotherapeutics, the science of treating the diseases of aging.
And that matters for getting these things approved.
It's crucial for regulatory legitimacy and insurance reimbursement. The FDA has defined pathways for treating specific age-related conditions like frailty or sarcopenia, but not for anti-aging. To achieve broad adoption, longevity treatments must be rigorously classified as medicine for specific conditions, not just lifestyle enhancements.
It's a strategic move.
It's a strategic move to ensure these technologies become available to the general public, not just the ultra-wealthy.
That makes perfect sense. Classifying aging as a treatable disease is the key to unlocking the regulatory gate. But as we manipulate these fundamental biological systems, we have to maintain a continuous safety watch. What are the long-term unknowns, particularly with the cell-based therapies?
That's a critical question. As powerful as these tools are, they are fundamentally altering biology. Gene editing precision requires meticulous, multi-decade monitoring for long-term off-target effects. And even with partial epigenetic reprogramming, PER, using the OSK factors requires rigorous surveillance to ensure there is zero risk of unwanted, latent pluripotency or oncogenic transformation down the road.
Right.
We are rebooting the system, and every reboot needs a failsafe and a full security scan.
Wait, how confident are we that this precise transient pulse is safe long-term? Are we sure we aren't introducing latent oncogenic risks that just haven't expressed themselves yet?
We can't be 100% sure, and that's why the research is starting in these contained high-need areas like the eye and the liver. The confidence stems from the fact that the factors are only expressed transiently. The pulse. The cells are not permanently exposed to the reprogramming signal. The goal is to induce the repair and then allow the cells to stabilize. If the trials show stability after that pulse, confidence will grow, but these platforms will require far longer safety follow-ups than traditional pharmaceuticals.
So, looking ahead, the path forward is clearly about convergence. We've seen powerful single-target solutions, but the future is combinatorial. What does that next generation of clinical strategy look like for the listener?
It's no longer a linear process. It's a stacked three-part protocol hitting multiple hallmarks of aging simultaneously.
Okay, what are the three parts?
First, you clear the garbage using robust immunological tools like Senovax or the anti-UPAR RT cells. Second, you repair the lost function using partial epigenetic reprogramming or engineered stem cells to restore that youthful code.
And third?
And third, you protect and optimize the system using metabolic regulators like microdosed GLP-1s and those AI-designed compounds. The true future of longevity is in stacking these validated approaches for a synergistic effect.
And finally, let's talk about that key basic science discovery. A fundamental, hidden layer of genetic control that the research team tracked. One that might dictate how these future drugs are ultimately designed.
This is a discovery that fundamentally refines our understanding of genetic control. Our intelligence highlighted studies showing that alternative splicing, which is the process by which a single gene codes for multiple different protein variants, correlates more strongly with maximum lifespan than simple gene expression levels.
So it's not just about turning the volume up or down on a gene, which is expression. It's about whether the gene is producing the correct version of the protein.
Precisely. Think of a gene as a blueprint. Splicing is the way the construction crew reads that blueprint, selecting which parts to include and which to discard to build a functional protein.
Okay, I'm with you.
As we age, or in shorter-lived species, errors accumulate. We call this splicing noise. The construction crew gets sloppy and builds compromised, truncated, or dysfunctional proteins, even though the raw materials, the gene expression volume, are adequate.
So the quality control fails.
It does. Long-lived species have evolved these molecular programs that optimize this splicing fidelity. They prevent the noise that accumulates in shorter-lived organisms. And the study found this fidelity effect was twice as pronounced in the brain, suggesting that splicing stability is the ultimate bottleneck for overall longevity and cognitive preservation.
This is truly a provocative conclusion. It suggests that the next generation of longevity drugs won't just adjust the volume of a gene, they will tune the complexity and precision of its output. The splicing fidelity, ensuring the cellular machinery is built to pristine specifications. The ultimate challenge that Tomorrow Unveiled will track may be solving this hidden layer of genetic control, allowing the human body to perform with the perfect software integrity of its youngest state. We love hearing your thoughts. Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about the Immortality update. Specifically, whether you'd sign up for a longevity vaccine or be interested in partial epigenetic reprogramming. Thank you for joining us on Tomorrow Unveiled. We'll be here next week, continuing to track the cutting edge of science and technology, separating the signal from the noise, and helping you navigate the future. We hope you join us then.